that audio is audible to you? Namaste.
Nikita, come to the second page. Yashwantini. One can occupy over here and one can do. Next to Shanmukhpriya. It is not working.
Hello, ma'am. Hello, students. Good evening. Hello, students. Good evening. Hello, students.
Namaste, sir. Am I audible?
Yeah, yes, ma'am. Yes, ma'am. Ma'am, I'm just downloading a question paper. Few minutes, ma'am.
Sir, am I audible, sir? I couldn't hear your voice.
Hello, ma'am. Yes, ma'am. Ma'am, am I audible? Hello, ma'am. Ma'am, can you hear me?
Select
Ma'am,
the
can you hear me, ma'am?
same as system and
Ma'am, am I audible?
NEET class ಅನ್ನೋದು NEET class ಅನ್ನೋದು
12 sir.
12 ಎಲ್ಲ ಶ್ರೀದೇವಿ
JE class போகுது.
NEET class சப்ஸ்டியூஷன் போட்டுருக்காங்க
இல்ல மேம் இது ரெடி பண்ணாதான்
அவரை இல்ல அவரை நான் வந்துட்டு அவரை எங்க
அது
நான் அவர்கிட்டமா அவர்
அப்படியே தப்பு.
எனக்கு இன்னைக்கு இங்க சப்ஸ்டியூஷன்
இல்ல அவர் எதுவும் பண்ண மாட்டாங்க
நான் அவரை
கனெக்ட் ஆகல
Hello, ma'am. Ma'am, can you hear me? Hello, ma'am. Can you hear me? Ma'am? Hello, ma'am. Can you hear me? Hello, students. Can you hear me? Hello. Hello, ma'am. Ma'am, can you hear me?
Sir, am I audible, sir?
Ma'am, am I audible?
I couldn't hear your voice, sir.
Oh,
Actually, we couldn't hear anything. What you
okay.
spoke.
Hello? Ma'am, can you?
No, ma'am, can you hear me now?
So you have muted the volume,
Hello, ma'am. Ma'am, can you hear me? Hello, ma'am, can you hear me? I'm just talking. Can you hear me? Hello, madam, can you hear me? I'm talking. Hello, hello, students. Hello, students. Can you hear me?
Full volume.
Hello,
Sound output இதுல தான் இருக்கு.
hello, students. Can you hear me? Hi. Hello, students. Can you hear me?
மேம் அவருக்கு ஆடியோ அவுட்புட் வருதா? இதுல அவுட்புட்டே வரல. மேம் அவர் பேசுறதே கேட்கலன்னு நினைக்கிறேன். இதுல அவுட்புட்டே வரல. இதுலயே அவுட்புட் வருது.
Hello, sir. Can you hear me? Hello, sir. No, I can hear you. Sir, I can hear you. Hello, sir. Where I can message?
இல்ல சார் அது இல்ல. நான் வந்து இந்த இதுல வச்சு இந்த இதுல இந்த ஸ்பீக்கர் தான் வர்த்தாதுன்னு சொல்லிட்டு இந்த ஸ்பீக்கர் இதுல
Sir is possible to hear. He is able to hear. We are not able to. I'll mute பண்ணிட்டேன். Unmute பண்ணிட்டேன்.
please check your audio sir.
I can hear them, sir. Please, but I can.
Audio அது proper.
இதெல்லாம் வேற HDMI கனெக்ட் பண்ணி பாருங்க.
Hello, sir. Can you hear me? Hello. Hello, students. Can you hear me? Hello, students. Can you hear me?
Students, is sir audible? Am I audible?
வேற ஏதாவது இல்ல அப்பதான் கம்ப்யூட்டர்ல ப்ராப்ளம் வந்து வேற ஏதாவது ஆடியோ கேட்குது.
இதிலே போடு பார்க்கலாம்.
Are we audible?
நாளை மட்டும் கரெக்டா போயாச்சுன்னா நம்ம சிஸ்டம் பார்ப்போம். Ashley, kita beda.
இல்ல இதுல ஆடியோ ப்ராப்பரா சூஸ் பண்ணுங்க.
இந்த ஆடியோ ப்ராப்பரா தான் சொல்லுது.
சேம் சிஸ்டம் போடுங்க.
ये कंप्यूटर ऑडियो
Students, are we audible now?
No,
மைக்ரோஃபோன்
Students, are we audible now? Yeah.
போடுங்க. அதுதான் சூஸ் பண்ணுங்க.
அதெல்லாம் இருக்கு.
அன்மியூட் பண்ண சொல்லுங்க.
लीव कंप्यूटर ऑडियो लीव कंप्यूटर ऑडियो கொடுங்க சார் லீவ் கம்ப்யூட்டர் ஆடியோ கொடுங்க கீழ ரெட் கலர் இருக்குல அது
கம்ப்யூட்டர் ஆடியோனா
இல்லதான்
அது சேம் தான். இதுதான் பண்ணுவோம் யூஸ்வலா. இப்போ அவர் அன்மியூட் பண்ணிருக்காரு. அவர் சைடுல கூட பிரச்சனை இருக்காங்கல்ல. ஆடியோ
sir we are audible now now we can hear you sir please sit in the chair you are going to connect that answer
Hello students, can you hear me now?
yes sir
Yeah, okay. And sorry for the delay, some kind of technical issues. Hope this chapter like biomolecules, okay, and very important chapter because not just for class 11, but in class 12, you people will be reading about like the molecular genetics and biotechnology. So there are two important units are there in class 12. One is molecular genetics and then biotechnology. So this unit is the basis of all those chapters, okay, about the molecules. Even in NEET, you get a lot of questions in this particular chapter, okay, and so hope you would have read this chapter, or else just have an idea. If the chapters are finished, no problem, you can answer. Otherwise, just have an idea how questions are asked from this chapter, okay. Fine. So we will I think I have some 50 questions, we will move as quick as possible. And sorry, I cannot write anything on the pad. Just I will be dictating you the answer key for question number 1. What is the answer key? So that part I will be telling you, okay. So please answer the questions if you know, otherwise we can skip to the next questions, okay. So read the first question carefully and tell me your answers. About 90% of the mass of every living organism is composed of just six elements. Okay, six important elements, including carbon, hydrogen, nitrogen, oxygen, and what are the other two substances? There are six important elements. Four are mentioned in the question. What are the other two? Yes, it is option D, calcium and phosphorus. Okay, it is calcium and phosphorus, option D. Okay, fine. Just keep in mind. And then the second question, enzymes, vitamins, and hormones can be classified into enzymes, vitamins, and hormones can be classified into single category of biological chemicals because all of these are.
Excuse me, sir.
Yes, ma'am. Yes, student. Yes.
Sir, this lesson was not took, sir. Biomolecules are not took, sir. Yes, sir.
Okay. So then you
Explain.
asked me some questions. See, listen. Cell over a cell. The fundamental unit of cell over.
Yes, sir.
Okay. Can I don't have any questions. Just like that, can I ask you some questions?
Yes, sir. You will try to answer, sir.
Okay. You try to answer, but I cannot present anything. Just like that, let me ask you some questions. Okay. Or else, if you have any doubt in cell, also you can what answer means you can ask. Okay.
Okay. Okay, sir.
Yes. So let me know what are your doubts in cell. Yes, tell me your doubts in cell. Okay, fine. What are the two type of cells? What are?
Eukaryotic cells and prokaryotic cells.
Very good. Okay, fine. Who proposed the cell theory? Which year the cell theory was proposed? Hmm? In which year cell theory was proposed?
1831, sir.
1838, 1839. Okay. What are the name of two scientists? Two scientists. Yes. Theodor Schwann, Schleiden, Matthias Schleiden, and Theodor Schwann. Okay. Omnis cellula e cellula. Statement given by C. Cell. Huh? Okay. Rudolf Virchow. Very good. Okay, fine. See, what is plasmid? In a prokaryotic cell, what is plasmid? Plasmid. Anybody can define plasmid? Okay. See, plasmid is chromosomal DNA or extra chromosomal DNA.
Extra chromosome DNA.
Very good. It is linear or circular.
Linear.
See, prokaryotic DNA or circular.
Circular, sir.
Yes, very good. Okay. How many chromosomes will be there in a bacteria? Bacteria contains how many chromosomal DNA? One, two, many,
One, two.
or many. How many chromosomes bacteria will have? Single chromosome. Okay, bacteria will have single chromosome. Okay, fine. What is the size of a bacterial cell? Common size of a bacterial cell. The student, size of a bacterial cell. Yes. Size of bacterial cell. Yes.
3 to 5 micrometer.
Okay, 3 to 5 micrometer. But what is given in the next page? If you look into the next page, a small typical bacterial cell size is there. Look into the picture in your book and tell me what it is given there.
1 to 2 micrometers.
So what is the correct one? It is 3 to 5 micrometer or 1 to 2 micrometer. There are two answers given. What is the correct one? Hello, students?
Sir, yes, sir. But also in book they have given that bacteria can be up to 3 to 5 micrometers.
Okay. Okay, fine. 3 to 5 micrometer. The smallest, see?
0.3
Okay.
micrometer.
Okay.
Microplasma.
3 to 5 micrometer because it is clearly mentioned there. What is the size of mycoplasma?
0.3 micrometer.
Okay, 0.3, 0.5 micrometer. Very good. Okay, fine. Now, what are the organelle present in bacterial cell? Inside the bacterial cell. What organelle is present inside the bacterial cell? See, when you see the structure of bacteria, no internal organelles. Internally, there is no organelle in the cytoplasm. Mostly, no membrane-bound organelle are present. Okay, no membrane-bound organelle are present in the bacteria. Okay, and what kind of organelle present in bacterial cell, inside the bacterial cell?
Nucleolus.
In bacteria,
Nucleoid.
okay, nucleus is the region that is not an organelle, that's just the DNA. Okay, keep in mind that's not a organelle, it's a DNA. Okay, next, see, ribosomes. Okay, ribosomes are the important organelle present in the, that is the organelle which is present in the inside the bacterial cell. And also, what is the infolding of membrane called? Very good. Mesosomes are the infolding of the membrane. Okay, very good. Fine. Bacterial cell wall. Okay, bacterial cell wall is made up of what? What is the component of bacterial cell wall?
Peptidoglycan.
Very good. See, what makes peptidoglycan? See, what are, listen, NAM, NAG. They call it as NAM, NAG. What are NAM and NAG? Hello, students. What are NAM and NAG?
N-acetylmuramic acid and
And
N-acetyl glucosamic acid.
very good. So they are the primary component of what? The peptidoglycan. Very good. So what are the two type of bacterial cell wall?
Gram positive, gram negative.
Very good. Very good. Gram positive and gram negative cell wall. Okay. What is the function of flagella apart from locomotion? Function of flagella apart from locomotion. Yes, locomotion is the function. Apart from that, what is the function of flagella? See,
To attach to
no.
the cell membrane.
Actually, it is called chemotaxis. Please note down. The function of flagella is called what? Chemotaxis. Means through flagella, if there is any food substances, okay, if there is any food substances, the bacteria senses that, a kind of consciousness, actually, consciousness. So bacteria, conscious about its environment. If there is a food, it moves towards the food. That is called what? Positive chemotaxis. And if there is a threat, for example, change in temperature, change in pH, which affects the viability of bacteria, the bacteria moves away from there. Okay? Positive chemotaxis and negative chemotaxis. Okay? Fine. Flagella function. See, what are the modes of sexual reproduction in bacteria? Modes of sexual reproduction in bacteria.
Through sex pillars.
Yes, sex pillars is there. There are 3 modes. What are they? Say, note down. Transformation, transduction, conjugation. Okay? The 3 modes of sexual reproduction in bacteria are transformation, transduction, conjugation. See, transformation means from the dead bacteria, DNA is transferred to the live bacteria. Okay? From the dead bacteria, DNA is transferred to the live bacteria. See, listen. In bacteria, you may confuse where this term is there. Okay, where these things are discussed. Before the paragraph of mycoplasma, you get all these things. Genetic recombination occurs in bacteria. So, transformation one method. And then, transduction means through virus. Okay, from one bacteria to other bacteria, through virus, DNA is transferred. That is called what? Transduction. Conjugation is, as you said, conjugation through sex pillars. Okay, conjugation occurs through sex pillars. Okay, keep that in mind. Very important questions. Next, coming to the prokaryotic cell, like when you compare mycoplasma with bacteria, what organelle will be missing? Okay, when you compare cell wall, very good, very good, fine. So mycoplasma is parasitic or what?
Can you repeat, sir?
Mycoplasma or parasitic or photosynthetic? Mycoplasmas or parasitic or photosynthetic? They are parasitic in nature. Okay, mycoplasma, yeah, parasitic. They can live on host like plants and animal cells. Okay, fine. Next, coming to animal cell. Okay, animal cell. Okay,
Yes, sir.
what is the size of red blood cell? What is the size of red blood cell? Hmm?
7 micrometer.
Okay, 7 micrometer. Good. What are endosymbiotic organelle in eukaryotic cell? What are endosymbiotic organelles in eukaryotic cell?
Endoplasmic reticulum.
No, actually, listen. Mitochondria and chloroplast are endosymbiotic organelles. Okay, mitochondria and chloroplast are endosymbiotic organelles. Okay, what is endosymbiosis? Actually, mitochondria and chloroplast are bacteria in early days of evolution. They are engulfed by, okay, eukaryotic cells. Okay, that's why we call them as what? Hello students, please listen. That's why we call them as what? endosymbiotic organelles. Okay, mitochondria and chloroplast are endosymbiotic organelles. Okay, and what are the molecules? Next question. What are the molecules which makes the cell membrane? What are the molecules which makes the cell membrane?
Phospholipids,
Yes, phospholipid.
cholesterol, protein, carbohydrates.
Yes, cholesterol, protein, carbohydrates. Okay, this composition was studied using what type of cell? Based on what type of cell they studied the composition of cell membrane? Based on what type of cell?
Erythrocyte,
Yeah, very good. Red blood cells, erythrocytes. Based on erythrocytes only, the composition of cell membrane was studied. Okay, and then the model of cell membrane proposed by whom? Model of cell membrane.
fluid
Hello.
mosaic,
Yes, fluid mosaic model proposed by proposed by
Nicholson and Singer.
yes, Singer and Nicholson. Very good. Now, what is active transport? What is passive transport?
Active transport means ATP.
Active transport means
It needs energy for
yes,
transportation.
it needs energy. Passive transport means
It doesn't need energy for transportation.
okay, fine. Uphill movement occurs in active transport or passive transport. Uphill movement
Active transport.
because why you call it as uphill movement?
Because it's against the concentration.
Means yeah, from low concentration to high concentration substances has to be transported. That's why it is called uphill movement. So passive transport is called what? Downhill movement, from high concentration to low concentration. Okay, fine. Now diffusion is active transport or passive transport?
Passive transport.
Diffusion.
Passive transport.
Passive. Okay, diffusion is passive transport. Okay, diffusion is passive transport. Okay,
Yes.
diffusion. Okay, listen. Diffusion occurs through what? Diffusion occurs through
Plasma.
channels, lipid bilayer, pumps, all of this. What is your answer? The question is diffusion occurs through channels, lipid bilayer, pumps, all of this. What is your answer? Okay, listen. No, very good. Listen. Diffusion is passive transport, so it can occur through lipid bilayer and channels, cell membrane channels. If it occurs through lipid bilayer, it is called simple diffusion. If the diffusion occurs through channel, it is called facilitated diffusion. Okay, simple diffusion, facilitated diffusion. Okay, through pump, only active transport can occur. Okay, through pump which are present on the cell membrane, only active transport can occur. Okay, fine. So, osmosis. What is endosmosis? What is exosmosis? Endosmosis and exosmosis.
The process where molecules move from high concentration to low concentration.
Okay, osmosis, endoosmosis, exoosmosis means what?
In endoosmosis, for example, water moves inside the cell. In exoosmosis, water goes outside the cell.
Very good. Okay, now when a cell is placed in hypertonic solution, what occurs? When a cell is placed in hypertonic solution,
Exoosmosis.
why?
Because there is high concentration inside the cell.
Yeah, very good. Solute potential is more in the extracellular fluid. That's why water can move from high water potential to low water potential. So if solute is less, water will have more potential. Okay, if solute concentration is high, water will have less potential. So in that condition, water can move from inside the cell to outside the cell, exosmosis. So many questions they may ask from this concept, endosmosis, exosmosis. Okay, and then hypertonic solution, hypotonic solution, okay, or isotonic solution. So tonicity, based on tonicity, they may ask lot of questions. Okay, next, what are the organelles comes under endomembrane system? What?
Endoplasmic reticulum,
Yes.
Golgi apparatus,
Hmm.
lysosomes and vacuoles.
Lysosomes and vacuoles. Very good. They are the endomembrane system. Very good. Give me examples of organelles with single membrane. Organelles with lysosomes,
Golgi complex,
single membrane.
endoplasmic reticulum, vacuoles, lysosomes.
See, listen. Lysosome and vacuoles alone will have single membrane. Nucleus, endoplasmic reticulum, Golgi bodies, okay, mitochondria, chloroplast, all these organelles will have double membrane structure. Okay, single membrane organelles in a cell, eukaryotic cell, if you study, single membrane structure and double membrane organelle. Examples you should know. Okay, fine. And then, what is the difference between cristae and cisternae? What is the difference? Cristae, cisternae. Cristae, cisternae. See, cisternae is present in endoplasmic reticulum and Golgi apparatus. The disc-like structure present in endoplasmic reticulum and Golgi apparatus are cisternae. The infolding of mitochondria is called what? Cristae. Okay? Cystae, cristae. That difference you should know. Okay. Where post-translational modification occur?
Can you repeat, sir?
Where, in which of the organelle post-translational modification occur?
Golgi body,
Very good. What is post-translational modification? What is?
after
Ah. See, okay. Translation means what? Translation.
change,
Huh?
change,
Translation means
transformation of proteins.
yeah. See, protein synthesis. Okay. Listen. DNA. Listen. What is replication? What is transcription? What is translation? Replication means what?
What, sir?
Replication means what? See, replication means what?
Xeroxing.
Yes, still listen. DNA makes the parent DNA makes two identical daughter DNA. Okay, DNA to DNA, one DNA, one parent DNA makes two daughter DNA. That process is called what? Replication. Replica means identical copies. One parent DNA produces two identical copies. Okay. Next,
Okay.
transcription means from DNA, RNA is synthesized. That to mRNA. From DNA to RNA synthesis is called what? Transcription. Okay. And then from RNA to protein is synthesized. That is called what? Translation. Okay, translation. So this is the central dogma of molecular biology. They call it as this process, DNA to RNA, RNA to protein. This process they call it as what? Central dogma. Okay, very, very important thing which you study in molecular genetics in class 1, but know where these processes occur. Okay, fine. Now, what is the pH of lysosome? Inside the lysosome, what is the pH?
Acidic pH 4 to 5.
Acidic, very good. 4 to 5, acidic pH. Wonderful. Okay. And then what make and what are made in smooth endoplasmic reticulum?
What, sir?
Smooth endoplasmic reticulum.
It doesn't have ribosome.
Okay. Smooth endoplasmic reticulum produces lipids, primarily lipids, lipid biosynthesis. Okay, fine. And then coming to the nucleus, okay, and chromosome. What are the four type of chromosome? Okay, still listen. Yeah, based on the position of centromere, metacentric, submetacentric, acrocentric, telocentric. Just one last question. Among these four type of chromosome, which is not present in humans? Yes, ma'am. Okay, ma'am. Anyway, see, telocentric chromosomes are not present in humans. Okay, all three types are present. Anyway, students, thank you so much, students. Thank you so much for your patience. Thank you.
Thank you, sir.
Yeah, thank you. Thank you, students. And the one who answered all the questions, special thanks for the student who answered all the questions. May I know her? Who is she? The one who answered all the questions. May I know her?
Trinity, sir.
May I know? Can you please come in front? I'm so glad to have you. Please, I would like to see you. Okay, thank you. All the best. Okay, Sri Devi, right? Thank you. All the very best. Cheers for you. Lot of
Thank you.
cheers for you. Okay, your support was so important. Thank you so much, Sri Devi. Sri Devi, right?
Yes, sir.
Yeah, yeah. Thank you. Thank you.
Thank you, sir.
Thank you all. Thank you, students.